exists in the bubble bear species of planet Pop. The typical dihybrid cross produces a phenotypic ratio of 9 to 3 to 3 to 1. Here we can see two bubble bear aliens. Each is heterozygous for loci A and B, as seen right here. The two genes interact to control their pigmentation. In a condition with no epistasis, we might experience 4 different phenotypes. A phenotype where we had big A and big B, big A, little b, little a, big B something, little a, small b, small b. This would give us that 9 to 3 to 3 to 1 ratio, and you can see that based on this Punnett square right here. The Punnett square has 16 different options, and from those, 9 give us one phenotype, 3 give us another, 3 give us another, and 1 gives us the other. However, in real life in the planet Pop, we actually experience only 3 phenotypes. Bubble people are either light pink, rose pink, or hot pink. Therefore, there must be some sort of epistatic interaction occurring. Let's look at the chart below to see which one it could possibly be. Since we have only 3 known phenotypes, it must be single recessive, single dominant, or duplicate genes with cumulative effects based on the phenotypic ratios. The only way we can tell what it's doing is by a test cross of the dihybrid parents. Here we have, in the test cross, we have parents with genotypes big A, big B, little b, and we produce 3 phenotypic types. We get a ratio of 3 rose pink, of 12 light pink, 3 rose pink, and 1 hot pink. This 12 to 3 to 1 ratio suggests that we have a single dominant epistatic relationship. Below is the biochemical pathway for the conversion of pigmentation in bubble bears. Single dominant epistasis occurs when the A something genotype results in some kind of repressor that prevents the conversion of the precursor to the intermediate, the precursor being light pink, the intermediate being rose pink. Those with the big B in their genotype will allow the second enzyme to convert the pigment to the hot pink stage. Finally, those with 2 little b's will stop the enzyme from converting the hot pink, and they will be stuck at rose pink. This results in the genotypes that had a big A combining to form the 12 to 3 to 1 ratio. Lastly, let's look at a test cross between 2 parents of the following genotypes. Based on our knowledge of the genotypes, this cross would produce only 2 phenotypes with a ratio of 1 to 1, light pink to hot pink.